Welcome everybody to the 2018 Interpreting Genomes for Rare Disease Variant and Gene Interpretation course. We're really excited to have this many people this time around. This is our second time doing this as the Center for Mendelian Genomics, and we're really lucky to have ClinGen co-sponsoring this event this year. So this lecture over the course over the next 2 days is going to have both lecture-based talks as well as workshop-based talks. The lectures are to give you the basics and the background that you need for the hands-on opportunities that will happen in the workshops. The goal is to have all of you feel more comfortable with both the filtration, analysis, and interpretation of genomes. So we really encourage you to ask as many questions as you need and to get out of this what you need. So before we get started, there's a couple logistical things. One, I should introduce myself. I'm Samantha Baxter. I'm a genetic counselor here at the Broad Institute, working with Daniel MacArthur and Heidi Ream at the Center for Mendelian Genomics. Navigating the Broad. You will spend the majority of your time in this room and on this first floor in the lobby. If you need to use the restrooms at any time, feel free to get up at any point. If you go past the security desk out here, on the left-hand side are the restrooms. There are going to be a couple times throughout the day for workshops that some of you may have to go to different floors. If you need to use the restrooms when you're on those different floors, just come ask one of the staff members. They're sometimes a little harder to find, so it's easier to show rather than describe. So just ask one of our team members and they'll show you how to get to the restrooms on that floor. All meals and the social event this afternoon will take place out in the lobby. For those of you who paid for the networking and social event, you will recognize that because you have an orange sticker here on your badge. If you feel that you did pay for this event and you don't have an orange sticker, please go to the registration desk and let one of us know. If you didn't pay but would like to attend, we can arrange for you to pay on site and we can give you an orange sticker as well. The lectures will all take place here in the auditorium, but some of the workshops will occur on other floors. Before we split up for the breaks before the workshops, I will tell you where you're supposed to go on a slide, so just try to take note of where you're supposed to head. But again, if you're not sure, you can come find me. Also, all of your workshops that you selected during your registration were based on your skill level. So we're going to break down the workshops off of your self-selected skill level. There's stickers on the back of your registration, and it tells you what workshop you selected. If after you hear the talks and you feel that that is not the appropriate level, you could always self-select to go to one of the other ones. That's not your final one. I'm just trying to remind you what you said you thought your skill level was. Your name badges will give you access to the first and second floor, so you will need to show them to security. But if you are going to any other floor other than the first and the second floor, you will need to get elevator access. So you will have to go with a Brody or somebody who has access to the floors, and we will take you up and down on those, or up on the elevators. You can get down. Now, I know there's a lot of people who like to use stairs. Do not take the stairs while you are here. You will get locked in the stairwell if you try to get out, and it can be rather embarrassing even from a Brody who forgets their badge sometimes. So just if you need to do laps, walk these stairs or walk around the lobby, but just it's better to not take the stairs during the time that you're here. For all of you, you will need Wi-Fi, so you can access our public Wi-Fi using Broad. There should be no password required. And now for actual course details, all slides and workshop materials will be located on the shared drive. This is the link. I will make it bigger on the next slide for those of you who can't see like myself. But you also got this shared drive in the confirmation email that was sent out last week. All talks happening in the auditorium will be recorded, which means we ask that if you ask any questions at all, that you come up to these two mics in the front. If you feel like you can't come to these two mics, please raise your hand and we'll bring the mics up to you. But we do want to have everybody's questions recorded, so we ask that you try to make sure you do that. If you are planning to collect CEUs, please be sure to sign in at the front desk each day. So you need to sign in both days. You should have all signed in today. You will also need to bring a check and fill out a survey at the end. So if you have any questions about that, please find Danielle or myself, and we can help you through that process. We are asking you to use your own computers during the workshop. You should have all received an email letting you know to bring your computer. If you did not bring your computer or your computer is not working for any reason, please find a friend during these workshops and come find us if neither of those solutions is possible for you. We will have disclosures throughout, but I just wanted to point out that none of our speakers have any conflict of interest to disclose. And finally, please ask questions, be engaged, and ideally have fun. So again, the Wi-Fi is Broad, and then I'll just take a second for any of you who need to write down or type in the folder. Make sure that you can connect to both of these because we will be using the internet and we will be using the shared drive throughout the presentations, or throughout the workshops, I should say. All right, looks like people have stopped writing, so I can. So I just wanted to take a second to thank some key people who were really helpful with this course. Danielle Azaridi, who's walking up the stairs right now, from ClinGen, who was a huge help in bringing this all together. Joanna Frenkel from Broad Events. She made all this look professional and looks like we knew what we were doing through the whole process, so I'd really like to thank her. And then Jamie Chang from our group at the Broad CMG. She made the website and maintained all of it flawlessly so that I just had to tell her what to do, and she took care of all of it. And without the website, most of you couldn't register, so she was very key to making sure this happened. So quickly, before I bring up Daniel to introduce the course and to kick off our first talk, I just wanted to remind everybody about our course objectives. These are used for the CEUs, and we'll ask you all at the end to let us know if we met these objectives. But we want to explain the process of next-generation sequencing data generation, apply the guidelines and best practices for variant and gene curation, and compare and contrast resources to support gene and variant interpretation process, including data sharing sites and functional databases. And with that, I will bring up Daniel MacArthur, who's the co-director of our Broad Center for Mendelian Genomics and the Medical and Population Genetics Group here at the Broad Institute.
Thank you so much to Sam for that introduction, and of course, thank you to Sam and the team for all of the hard work they have put into putting this two-day course together. And I also want to say thanks to all of you for traveling out here to visit this. And I know some of you have actually come a long way to get to this course. We're looking forward to the next 2 days. This is obviously the first time we've thrown this. We look forward to interacting with you and learning about what you are interested in getting out of the course. But we'll also be very interested in hearing from you about what we've done well, what we haven't done well, and how we can improve things the next time we run it. So my goal in this starting presentation is to give an overview of next generation sequencing methods. I'm going to start by talking about how we potentially select cases for sequencing, and in particular, I'll give you some background on the approach that we take in the Broad Center for Mendelian Genomics that Sam already mentioned, and what other considerations we factor into designing studies for sequencing, studies and sequencing approaches for rare disease patients. We'll spend a little bit of time, but not going into a huge amount of detail, about how the process of next generation sequencing actually works. And then I'll do a walkthrough of technologies with a couple of examples of some of those technologies, specifically exome, whole genome, and RNA sequencing, again, giving some insight into the strengths and the weaknesses of each of those different strategies, so the particular situations where we might actually use each of those technologies. But before we start getting into technologies, it's worth acknowledging that human disease is an enormously diverse set of things that we need to wrestle with. Diseases in general fall on almost all diseases have some genetic component, but they fall on a very large range of genetic architectures. So on the one hand, on the left-hand side, we have diseases that are the diseases most of us here in this room work on, that is rare diseases, which are often very severe and monogenic. So these are things like Duchenne muscular dystrophy, for instance, where we have clear inheritance patterns within a particular family. Typically, there is only 1 or 2 causal variants per family that fully explain that particular disease, or largely explain that particular disease. And the causal variants are typically Mendelian or monogenic. That is, they segregate predictably within the family, although that's not always the case. And in this particular family, in this particular pedigree, of course, everyone here in this room will acknowledge this is a fairly straightforward-looking dominant disease pedigree. Then at the other end of the spectrum, we have common complex diseases. These are things like type 2 diabetes, schizophrenia, rheumatoid arthritis. These diseases are what we call massively polygenic. That is, in any one individual case, they're caused by a contribution of many different genes, often each of them with tiny effects, along with environmental factors that add up to cause that person's risk of a particular disease. And in general, the causal variants that are associated with these diseases tend to be much more common and have tiny effect sizes. So they're very different from what we tend to see in the context of the rare diseases we'll be focusing on over the next 2 days. And in addition to that spectrum of genetic architecture, there's also a wide spectrum of frequency and effect sizes for the genetic variants that underlie these diseases. So in this plot, we're showing, this conceptual plot, I should say, we're showing on the x-axis the frequency of the genetic variants that may underlie a particular disease. On the y-axis, we have the effect size, which varies from very low effect sizes, the types of variants that may fall out from a genome-wide association study, for instance. And up here, we have very large effect sizes, an odds ratio of 50. This is something that we might consider to be Mendelian. That is, basically everyone who carries that particular variant actually goes on to get that specific disease. So variants can, as I mentioned, fall along a spectrum both of frequency and of effect size. In general, there are almost no variants that fall into this section out here. That is, there are no variants that are both common and also have very large effects on disease risk, and that is because of natural selection. If there was such a variant that caused disease with a very high risk in a lot of people, it would generally be removed from the population by natural selection. There are many variants that fall in this category down here on the bottom left. That is, variants that are both extremely rare in the population and also have small effects. But in general, we have no statistical power to detect these variants, and so we don't know many examples that fall into this category. So in general, the variants that we find in studies of human disease fall along this diagonal line here. They range from, down the bottom right-hand side, the common small effect variants that are typically identified in the context of genome-wide association studies for common disease, and ranging right the way up through to the variants we'll be focusing on for the next 2 days. That is, very rare variants with extremely large effect sizes that result in what we would consider typical monogenic or Mendelian disease. And it's those variants that our group focuses on discovering, and in particular we do this through an effort called the Broad Institute's Center for Mendelian Genomics. This is a center that was funded and founded back in early 2016 to discover the new genes underlying Mendelian disease. The goal of this center is to work with collaborators, so we interact with people all around the world, including a number of people here in this room, to build up cohorts of patients consented for genetic studies. These are all patients presumed to be affected by severe genetic disease, and almost all of those have been pre-screened using panels or other approaches for known genetic causes of those diseases. We then cover the costs of exome and in some cases genome or RNA sequencing. We also support analysis, and we've analyzed over 2,500 families to date. I'll be talking about some of the things we've discovered over that course in the rest of this talk. And then we take those diagnoses and those gene discoveries and give them back to collaborators to do further pursue them and pin them down functionally and clinically. And one of the key components of the CMG is this strong commitment to data sharing. That is, we believe that it is in the best interests of the rare disease community as a whole that the data that we generate be made as quickly and as openly available as possible, and that's something we try to permeate through all the studies that we do. So our general strategy in the CMG and in other rare disease studies that we engage in is to identify patients who have often undergone a very long diagnostic odyssey and typically had substantial pre-screening for known genes associated with their disease. This is actually one of the first families we studied, a family with 2 young children, the only 2 children in this family, affected by severe congenital myopathy, resulting in severe facial weakness, as you can see. And in these families, the first thing we do is to sequence exomes. And exome sequencing, and I'll take a bit more time to talk about what that is in a minute, exome sequencing remains our workhorse technology. That is the technology that we apply across by far the largest number of samples, and it's typically our frontline approach before we start thinking about other technologies. We will typically sequence the parents in the family and all of the affected siblings that exist within a particular family. In the case of dominant pedigrees, we will sequence all affected individuals, the most distantly related affected individuals, and then sometimes also perform linkage across that entire family as well. So with that family exome, what we typically find is that we are able to identify causal variants in around about 25% of the cases that we analyse, although that varies wildly by disease cohort. It's much higher in some disease cohorts and substantially lower in others. And that depends, of course, on how much pre-screening has been done in that particular cohort and how genetically challenging that cohort is for analysis. And I'm sure we'll spend a little bit more time later talking about the reasons for those differences. We pursue lots of different kinds of analysis of our exome sequencing data. So we'll go back and revisit exome sequencing data over time, particularly as new genes are discovered. We can go back and look to see whether those new genes show up in our particular families. We'll also go back and apply new analytical methods, such as, for instance, looking for copy number variants that can be discovered using exome sequencing as well. And then for cases that continue to be resistant to being solved or diagnosed using exome sequencing for very long periods of time, we'll then consider taking them forward to use other technologies that I'll talk about later, like whole genome or RNA sequencing. So again, before we get into the technologies, I wanted to spend some time thinking about how we pick the types of families that go into these types of studies, because this might be relevant to the way you guys are thinking about your analyses as well. And the families we choose depends very largely on the goals of our work. That is, in particular, whether we're primarily focused on getting a diagnosis for as many families as possible in a particular project, or whether we're focused on the discovery of new disease genes, so genes that hadn't previously been associated with that disease. So if we're primarily interested in diagnosis, then things like pre-screening or having samples that have already been sequenced using a panel become much less important. What we're primarily interested in doing is taking families that come in from any background where they have clear evidence of a genetic disease, and then applying exome sequencing immediately to try to learn the basis of that disease. In general, if we're money limited, and let's face it, most of the time we are all money limited, we tend to favor exomes. But in some situations where the primary limiting factor is samples, that is, we have a particular disease where we have a very small number of samples with that specific disease, and we are desperately interested in learning the cause of that disease as quickly as possible, then we may go directly to whole genome sequencing, and I'll talk a bit more about that later. The situation is slightly different if we're focused on gene discovery, because here we're most interested in pre-removing families who have mutations in known disease genes. So here we tend to favor families that have gone through long odysseys where they've had lots of panel sequencing, lots of targeted gene testing done to rule out known causes of disease. And of course, we also tend to focus on clinically homogeneous families, so particularly large cohorts of families where where they all appear to be affected by a very similar type of disease. We focus on what we call solvable family structures, and I'll talk a bit more about that on the next slide. So that is families where it's more likely that the genetic cause will be solvable using sequencing approaches. And for gene discovery, we will almost always focus on exome sequencing, because the discovery of coding variants that underlie those particular diseases is most cost effective by applying this technology to large numbers of patients. So we look at a whole range of different family structures in our center. And one of our favorite types of families to look at, of course, is families where it's unambiguous or close to unambiguous that the underlying genetic cause is actually recessive. And that would be a family that looks like this, for instance, where we have two affected siblings with unaffected parents, and where really the only possible models for this are either a recessive cause that has happened to be inherited in both the children, or potentially a germline mosaic in one of the parents where a de novo mutation has been passed down to both of the children. But our primary family structure, of course, is trios, and so these are most of the families we study. We have a single affected individual with unaffected parents, and we will bring those in and perform exome sequencing across the entire trio in those cases. We have some number of small dominant families. We tend to focus less on these because these families are very hard to solve in general using exome sequencing approaches. When the focus is the discovery of new disease genes, it's very unlikely we'll be able to discover a new disease gene using these small families. And then a final study design is a large collection of probands all affected by the same disease. And so here we will tend to focus on cases where we can get access to dozens, ideally hundreds or even thousands of cases of a particular rare disease where we think that there is some kind of clinically homogeneous entity across that entire collection of patients. There's a whole host of other considerations for choosing samples that go into this type of sequencing approach. So of course, we need to make sure we bring in families that have the appropriate consent or IRB approval. We look to make sure that there's other family members, DNA samples available from other family members that we can use for follow-on clinical and segregation studies. We typically focus on families that have highly unusual clinical presentations, so things that are unlikely to be explained by the known genes. And wherever possible, for reasons I'll explain later, we look for families where there's an availability of cells or appropriate tissue samples that we could use for RNA sequencing, which can be really helpful for identifying variants that result in a disruption of splicing. So before getting into the sequencing approach itself, some cautionary notes about which factors we have found tend to favor success in diagnosis and gene discovery. The first really important note, and that has already come up in the slides on genetic architecture, is that it's really important for these studies to work that the disease actually be monogenic or close to monogenic. That is, it's caused by one or a couple of very large effect genetic variants that fully explain the disease in that particular family. Small family-based analyses are typically wildly underpowered for anything else. So that is, if you're interested in diseases like celiac disease or type 2 diabetes, you will not discover the underlying causal genes by sequencing a small number of trios, no matter how extreme typically the presentation is in those families. A second caveat that we have learned the hard way in sequencing our families is to be very wary of the results of prior screening. The accuracy of prior screening depends very heavily on who actually did that prior screening, and we actually identify when we apply exome sequencing in our families, even heavily panel screened families, we often identify causal variants falling in genes that have nominally already been sequenced as part of those panel testing. So there are still gaps in a lot of the panel testing that's currently being done. And a final note is the value of matching as closely as possible the types of technologies that have been used to sequence large reference databases like EXAC and NOMAD, which Anne will be talking about in her presentation later. Okay, so next, a really high-level conceptual overview of how the process of next-generation sequencing works. And at the highest possible level, you can think about this as basically taking a DNA sample, the entire genome of a particular individual, and smashing that up into really small fragments, and then reading each of those fragments. Those fragments vary in length. They're typically substantially longer than the fragments I'm showing on this particular cartoon, which is for illustrative purposes only. But here we might have, for instance, a 100 base pair fragment up to 300 base pairs, depending on the exact technology that's being used, even longer for some long-read sequencing technologies. So then we have millions or billions of these small fragments, and we need to then somehow use those to figure out exactly what genetic variants are present in that particular individual. And the way we do that is by looking at something called a reference sequence. You can regard this as a kind of average sequence that's been defined for the human population as a whole. And this is basically the reference sequence that all of us line up all of the reads against for all of the samples that we sequence. So we take that reference sequence, we take all of the fragments of DNA that have been obtained from our specific patient or individual, and we then use an algorithm like BWA to align those against the reference sequence. So this is basically taking each of those snippets and then placing it in the appropriate position along the reference sequence. And then our algorithm then scan along that reference sequence and look for positions where that patient differs from the reference. And so you can see here at all of these other positions, the reads perfectly match the reference, except at this position here. The reference here is a C. But at this particular position, this person has 4 C alleles, 1, 2, 3, 4, and 5 alleles that match a T. So that tells us that this person has roughly 50-50 dosage of a C to T polymorphism at this site. That is, this person is heterozygous for that C to T polymorphism. So that makes it all sound very simple. Of course, there are challenges in going from the short read data we get from these individuals to this collection of variants. Some of the challenges that we face in our analyses include the fact that these short reads may not map unambiguously to a particular place on the reference. This is especially true for repetitive regions of the genome, so it can be very hard to untangle whether a particular read is meant to map in one place versus another. Secondly, this is particularly true for exome sequence, as I'll show in a minute, but also true for genome sequence. There can be variable coverage across different sections of the genome. So we may find some regions that have really high coverage and some genomes where there's insufficient reads piling up in that particular region to get a good sense of that person's variation. Base calling quality can also vary from site to site. Base calling quality here is how confident we can be that the reads that pile up in a particular location actually very confidently tell us the precise sequence of that location. And that base calling quality tends to be worse on average for insertions and deletions compared to single nucleotide variants like this C to T mutation here. And finally, a particular challenge that I'm not going to go through in any real detail today, although I'll show you one example of how hard this can be later, is the fact that large rearrangements, that is deletions, large insertions, or large rearrangements of DNA can be particularly difficult to call from short-read sequencing data, basically because they tend to fall in repetitive regions, and reconstructing them from short-read sequencing data is often very challenging. So without going into the details of what each of these different file types look like, there are a set of different data types that you may well encounter over the course of your interactions with the next generation sequencing data, and these may well come up over the course of the next 2 days as well. So I want to just briefly explain the differences between these. So really there are 3 different classes of file that are important for next generation sequencing data. The first class of file is the sort of file that gets produced at the end of a sequencing run. This is called a FASTQ file, and it basically stores all of the reads that have come off that particular sequencing run, that is all the millions or billions of little snippets of DNA that I showed in that slide a couple of slides ago, and they're stored unsorted. So this is just a collection of everything that's come off that sequencing run. We then apply an alignment algorithm, and the second type of file that we create is called a BAM file, or typically what we store now is a compressed version of a BAM file called a CRAM file. This is a much more efficient way of storing BAMs. And a BAM file is also very simple. You can just think of that as the same collection of reads that you would find in a FASTQ, but now they're lined up against the reference genome. So for each of those reads, we know where that fits against the specific reference genome. And then we can take that BAM or CRAM file and identify the specific sites where there are variants in that particular person, and that then results in a variant call file or VCF. And that VCF simply lists for every single position in the genome where that person has a difference compared to the reference. It gives you the position of that particular difference, tells you what the sequence of that difference is, and then it gives you all kinds of information about that difference, like how confident we are that the variant is actually real, as well as optionally information about its functional impact and other types of information that might be useful in making sense of that particular variation. So these are the only real file formats that you're likely to encounter over the course of your interactions with next-gen data. Now, Anne will talk much more about this in her presentation, but I wanted to briefly mention that, of course, in order to make sense of the list of variants that we identify in a particular patient, we need to be able to line that up with a large number of other reference individuals, ideally tens of thousands or even hundreds of thousands of people who've been sequenced from the general population. And the reason that's so important is because the frequency of a particular variant that we find in a patient is one of the most powerful clues to whether or not it is actually disease-causing, that is pathogenic. And so ideally what we'd like to be able to do is to take each of the variants we find in one of our patients, look that up in as large a collection of sequenced individuals as possible, find out how common that variant is in different populations, and then if it is too common in one of those populations, we can almost always confidently rule it out as being pathogenic. If it's very rare, then it has a potential chance of being pathogenic. And as these databases get bigger and bigger, we start to be able to think about taking variants that we find in those large populations and looking to see whether those carrier individuals also have the same disease. And that, of course, is an important part of data sharing activities, and we'll talk more about things like matchmaker exchange later. So this requires very large cohorts of reference samples, and that is dependent on resources such as the ones that we've been building here at the Broad, like EXAC and Nomad. And again, you'll hear more about those from Anne later on today. Okay, so let me then finally talk about the 3 different technologies that we tend to employ in understanding the variants that are present in patient DNA and in making sense of that data in the context of RNA sequencing. Before I go into those technologies, here's a cartoon of a region of the genome with a protein coding gene spanning across it. Each of these grey boxes here is an exon, a protein coding exon of that particular gene, and the thin lines in between represent an intronic sequence. And so all of you, of course, will remember your basic molecular biology. This strand of DNA is translated into RNA, a long strand of RNA, and then those introns are clipped out, and the exons are then joined together to create messenger RNA, which can then go on to form the template for the development of the translation of a specific protein. Now, there are lots of technologies we can use to analyze genome and to analyze the variation that's present in the genome. One of them that we won't be talking about much today or tomorrow is genotyping arrays. Genotyping arrays give us information at only a relatively small subset of the sites throughout the genome. So there might be hundreds of thousands or potentially over 1 million different sites throughout the genome. And these are typically focused on common variants that are present at greater than 0.5% frequency in the population. And these genotyping arrays are typically useful in the context of rare disease in the sense that they provide us with information about copy number variants, that is large stretches of DNA that may be deleted or duplicated and that we can spot by looking at the intensity of the signal at each of these different probes. But these genotyping arrays miss rare variation entirely. So any genetic variant with a frequency of less than 0.5% typically is completely invisible to these arrays unless it happens to be one of those very large copy number variants. The second technology that you'll hear much more about, of course, over the next couple of days is exome sequencing. This is cheap. This slide is actually wildly out of date. The Broad will now sequence exomes for about $160. So exome sequencing is very powerful and cost-effective technology for analyzing the protein coding regions of the genome. So again, just these bits shown in red here, and those are the only bits that exome sequencing targets, and perhaps a small number of bases on either side of each of those regions. The way exome sequencing works is that people who have designed exome capture arrays have designed particular probes that pull up particular regions of the genome. So here we have, for instance, in red, each of the exons in a particular region of the genome. We design beads that contain probes that target those particular exons, and so only those exons stick to the outside of those beads, and everything else doesn't stick to those beads and is just floating around free in solution. We can then spin that out in such a way that all of the non-specific DNA gets washed away, and we're left just with the reads that bind to the protein coding exons of the exome, known collectively as the exome. And you can see here in this particular snapshot for this gene down here, for each of the protein coding bits of the exome, of the protein coding regions of the genome, we have these nice peaks of coverage corresponding to many, many reads that cover that particular exon. And exome sequencing, as the technology has developed, has become more and more specific and more targeted to particular protein coding regions, becoming much more efficient at pulling out those regions of the genome. So the benefit of exome sequencing is it gives us high coverage of those protein coding regions, but the coverage is also fairly non-uniform. And this plot is actually one of the best ways I've seen of illustrating this. On the x-axis, we have each of these dots here corresponds to an individual exon across the genome, a protein coding exon in the genome. The y-axis here corresponds to the number of reads binding to each base on average of each of those exons. So this really gives us a sense of how confidently covered each of those exons is. And you can see that the mean coverage is around about 70 to 80x, but the variation is enormous. So some exons are really highly covered, some exons are not covered at all. So you can see this little cluster of exons down here. So that means that exome sequencing gives us a lot of information about particular protein coding exons in the genome, but it may completely miss the information in others. Now that's in contrast to genome sequencing. This is a comparable plot for genomes. And genome sequencing, you can see, gives us lower coverage on average. So this is a good genome, about 40x coverage. But the distribution of coverage is very, very uniform. So you can see these are all the protein coding exons in the genome. You can see that most of these are covered with a coverage that is very, very close to 40x. So that's a big advantage of genome sequencing. So given that variation, why do we still do exome sequencing as our first line approach? There's lots of good reasons. The first is that it's cheap. So at current prices, we can do easily a trio family exome for the price of a single whole genome. In fact, in the prices that we're looking at right now, the differential is more like 6 times difference between exome and genome sequencing. Secondly, there is actually higher coverage on average in exomes compared to genomes, and that means that in sites that have very high coverage, we're more likely to be able to discover potentially causal variation in that particular region of the genome. An important thing about exomes is that it captures the bits of the genome that we actually know how to make sense of. So those protein coding regions, although they only cover 2% of the genome in total, include almost all the pieces of the genome that we know exactly how to make sense of. That is, if a variant falls in those regions, we can say whether it is a missense variant or a synonymous variant, and also make some better estimate about whether or not it's disease-causing. So that's very useful. And our tools for interpreting non-coding variants remain very limited. And a final important thing is that it actually captures most rare disease mutations. It's difficult to get a really good estimate of this number. We think certainly over 80%, likely over 85% of all disease-causing mutations in very severe monogenic diseases fall in or near protein coding regions. So the vast majority of the variants that we care about are actually captured really well by exome sequencing approaches. So the next technology we can look at is whole genome sequencing. So whole genome sequencing, as the name implies, is basically looking at all the DNA, so including both protein coding exons and the non-coding bits, that is the introns as well. So whole genome sequencing is substantially more expensive. The price remains over $1,000. It does sequence almost all of the 3 billion bases of DNA, except the pieces of the genome that are so repetitive that our short-read approaches can't actually get to them yet. And that's still about 5% of the genome. A really important advantage of whole genome sequencing data is that it allows us to discover copy number variation and large-scale rearrangements that we could miss using exome sequencing data. And it also allows us to, of course, identify non-coding variants, that is, variation in bits of the genome that are not captured using exome sequencing. But the challenges I mentioned in the previous slide of these is that most of the non-coding variants we can't make sense of. That is, we don't know what effect those might actually have on someone's risk of disease. But to illustrate the advantages of whole genome sequencing, I just wanted to give you one very quick snapshot of a case where we were able to solve using whole genomes that could not have been solved using exomes. This is a case of a patient with Duchenne muscular dystrophy. We actually have studied a substantial number of boys with Duchenne muscular dystrophy, all of whom have not been diagnosed using the standard approaches. And so a quick bit of background on this disease. As many of you will know, Duchenne muscular dystrophy is the most common form of muscular dystrophy. It's X-linked, so that is the causal variants are present in boys. They're effectively they're hemizygous, that is they're effectively homozygous. The phenotype of Duchenne muscular dystrophy is extremely distinctive. So the boys tend to have an age of onset around about the age of 4 or 5. They suffer a very rapidly progressive degeneration of their muscle. And critically, we actually have a really good biochemical test, that is the absence of dystrophin, that is effectively diagnostic for Duchenne muscular dystrophy. So we will often know that there are boys who definitively have Duchenne muscular dystrophy. They must have a mutation in the underlying DMD gene, but in a small fraction of those cases, we can't actually identify the causal variant, even after exon sequencing plus MLPA, an approach to discover copy number variants across the exons of that gene. So we took one of those boys that hadn't been diagnosed using standard approaches and applied whole genome sequencing to that boy, and we discovered a region of the genome that looks like this. You will see snapshots that look like this later on during the course. So for those of you who aren't used to looking at read data, this is a snapshot of a particular region of the genome using a tool called IGV that allows us to visualize the individual reads in a BAM or CRAM file. So remember that intermediate file format I told you about earlier. Each of these gray regions down here is a particular read. Up here you can see the exons of the DMD gene. This squiggle plot here shows you the coverage across this gene using whole genome sequencing data. And in this particular, here we're zooming in on one small region here, this yellow box. And you can see here there's an exon of the DMD gene right here. This patient is missing a chunk of intronic sequence, this region of about 3 kilobases about here. And then this region over here has something interesting happening that's highlighted by these different light blue and dark blue reads. And we were able, by staring at that region with sufficiently long and applying a number of different tools, to determine exactly what had actually happened to this person's DMD exon. And this is the result. What has actually happened is there's a small region of the genome that contains this exon, and what this patient has undergone is a deletion of about 3.5 kilobases of sequence up here in the intron, a very small deletion of 42 base pairs down here, and this bit in the middle here containing the exon has been flipped around. So there's an inversion of about 4 kilobases, and that puts the exon now in the opposite orientation. So when that patient's DMD gene is being read, that exon is no longer available to be spliced in to that particular exon. That exon is absent, and that results in a frame shift, the complete absence of dystrophin, and therefore that patient goes on to get a severe Duchenne muscular dystrophy. So this is important in the sense that this is a mutation that could not be discovered using any of the other approaches apart from whole genome sequencing, because the exon is copy balanced, that is, there is no actual loss of material. So copy number variant assessments that focus on the exons won't pick that up, like MLPA, for instance. The only deletion that we have here is intronic, and instead we have this flipping around of sequence here that can be relatively easily picked up using whole genome sequencing, but will be missed using other approaches. So that's a success story. Sorry, let me try to resurrect this with less scrambled text. Okay, that's better. So that's a success story from whole genome sequencing, but of course whole genome sequencing won't always work, and I think it's important to emphasize that, particularly to this crowd. There are many reasons why exome sequencing fails, and many of those failure modes won't be addressed by whole genome sequencing. So for instance, if the variant is present in your sequencing data, so there is a missense variant, for instance, but it's misannotated, you don't actually know that it's causal, the fact that you've done a whole genome sequence won't give you any better information about whether that variant is causal or not. If the variant is in a really hard to call region, so for instance, it's in a very repetitive region of the genome that is difficult to analyze using exome sequencing data, many such regions will also be very hard to reconstruct using whole genome sequencing. If the genetic cause is not monogenic, that is, it is the result of multiple mutations in multiple genes, again, the fact that we have genome sequence versus exome sequence data will not change our ability to solve that. And of course, if the inheritance mode is misspecified, again, that's not going to change your outcome if you do whole genome sequencing. So in our hands, whole genome sequencing, although it does give us better coverage of the protein coding regions of the genome and does allow us to find copy number variants, it only gives us about a 5 to 10% bump in the diagnosis rate compared to exome sequencing. And that is why at the moment we continue to focus on exome sequencing as our workhorse technology, both for diagnosis and for gene discovery. So the final technology that I'll talk about fairly briefly today is RNA sequencing. And this is an approach that allows us to look at that final product of the second to last product of a protein coding gene, that is the messenger RNA. So for RNA sequencing, what we do is we take all of the RNA that's expressed from a particular patient's tissue, and again, we smash that up and extract RNA-seq reads, and then map those back to the genome to understand the coverage and the splicing of particular gene exons of a protein coding gene. RNA sequencing is still somewhat expensive, so it's closer to $500 now for reasonably high coverage RNA-seq from patient tissue, but it's very different from DNA sequencing in the sense that you have to get the right tissue. So when we're doing DNA sequencing approaches like exome or genome sequencing, you can get patient DNA from blood, you can get patient tissue or cell lines, and in most cases, unless you're caring about somatic mutations, all of those DNA samples will be equivalent. It doesn't matter which one you sequence, you'll get the same answer. For RNA sequencing, that's very different. It really depends on which tissue you sequence as to whether or not you get the same answer. So importantly, RNA-seq only sequences the expressed regions of the genome in that particular tissue. So that's why it's especially important to get the right tissue. And it finds changes both to gene expression and to splicing. That is the process by which one exon gets joined to another in creating messenger RNA. So at a very high level, it's really important to pick the right tissue. If you're working on a muscle disease, you want to make sure that you sequence muscle, if at all possible. And obviously for some diseases like neurodevelopmental disorders, given the absence of fetal brain biopsies for the vast majority of patients that we study, it's going to be very difficult to get the right tissue. So this is an approach that is very powerful in the right diseases, but can be really difficult to apply in others. You need really well-matched reference data sets of RNA sequencing. So in the same way that we use GNOMAD and EXAC for DNA sequencing data, you need good reference data sets for RNA-seq. We use a project called GTEx for that approach, and you'll hear more about GTEx later. And importantly, RNA sequencing data are much harder to interpret than DNA. The tools for analyzing this are not as well developed, and so it typically requires a very careful pipeline and an analyst who really knows what they're doing with RNA sequencing to make sense of that data. If anyone is at the point of thinking about analyzing RNA sequencing data, I'd encourage them to check out, we have a paper that we published a couple of years ago on applying RNA-seq to Mendelian muscle disorders, and there is a full tutorial that you can follow at that link there, or drop me an email if you want to grab that URL later. So just very briefly to illustrate the power of RNA sequencing, I wanted to give you one example. This is a little girl, one of two cases that we studied a couple of years ago with undiagnosed collagen myopathy. So she, as you can see, she's wheelchair bound. She had pretty classic signs of some sort of collagen myopathy, suggesting that she had disorders. She had a mutation in one of the three chains of the collagen 6 gene. But she and the other patient that we were studying in this particular case series had undergone really extensive testing to look for that causal mutation. She had had clinical sequencing of all three of the collagen 6 genes. cDNA sequencing had been done from fibroblasts from those genes. She had deletion and duplication testing done actually across the entire genome with no obvious discovery there. Exome sequencing was then done, and we had then done whole genome sequencing, and none of those had revealed the causal mutation in this particular girl or the other case in this series. So we then applied RNA sequencing. We got a muscle biopsy from these two patients and applied RNA sequencing. And in one region of the collagen 6A1 gene, we got a result that looks like this. And just to orient you, basically what this particular plot is showing is each of these boxes here, you can see from these black boxes here, is an exon of the collagen 6A1 gene. These histograms here represent how deep the coverage is for each of the bases of that exon along that particular region. And these loops here tell us how many reads are indicating splicing from this exon relative to that exon. So in this control individual down here, this is an individual from the GTEx project, so someone who doesn't have muscle disease. You can see there are 3 exons in this particular region. All of them are well covered, and there's this really nice looping effect where you have clean splicing from this exon over to this exon, and then clean splicing from this exon over to this exon. Now, remarkably, in both of the collagen 6, the undiagnosed collagen 6 patients that we were sequencing in this series, we found something very different. About half of the reads were looping as in the controls, and the other half were looping into this region in the middle here, this brand new exon that was appearing in the middle of an intronic sequence that we had never seen before and didn't seem to be part of the normal sequence of the collagen 6 gene. So we want to try and figure out what was going on here. We went back to the whole genome sequencing data and looked at the sequence in this region to see if we could figure out what was going on to result in the splicing in of this extra exon. And it turned out both of the patients, remarkably, had exactly the same de novo mutation. It was a mutation in the middle of the intron, and it resulted in a C to T change that actually creates this GT donor splice site. So there's a brand new donor splice site in the middle of an intron. It just so happens in this particular region of the genome, there is another region up here which has an AG, so that is an acceptor splice site. And so with the creation of this new mutation, suddenly there was a new spliced exon that was appearing in the middle of this person's intron, creating a completely gibberish sequence that was being jammed into the middle of the collagen 6 gene. This results in an in-frame inclusion where there are now 24 amino acids, basically random amino acids that are shoved into the middle of the collagen 6 gene, and that actually falls immediately in the middle of a known critical domain. That is a glycine repeat domain that is known to be key for the binding of the 3 different collagen 6 subunits to create functional collagen. So this is a really interesting example in the sense that it tells us discovering this particular mutation, we had seen it on whole genome sequencing data, but we couldn't make sense of it. It just looked like any of the other non-coding mutations that could be found anywhere else in the genome. It was very hard to predict that this mutation would actually result in a brand new splice site being created. But with exome sequencing, of course, would have missed this entirely because it's out in the middle of non-coding sequence. And it's really only with RNA sequencing data that we were able to zoom in on that particular region and identify exactly what the causal variant was that was causing this patient's disease. So again, this is a case where this particular technology turns out to be really transformative from identifying the particular cause. And I just wanted to mention, we've now identified exactly the same de novo mutation, remarkably, in 30 other cases. It's been de novo actually in more than 15 families where parents are available. So this same de novo mutation seems to be happening again and again in that intron, and every time it occurs, it results in this new exon being created that results in this severe collagen myopathy. So when is RNA-seq relevant to analyzing patients? We think it's most useful in situations where you have as in this case, a really strong candidate gene or set of candidate genes, but you've done whole genome sequencing, you've taken other approaches, and you haven't been able to find the cause of that particular patient's disease. So a great example here might be if you have a recessive gene that has a single hit, you found that single hit, but you can't find the second hit. RNA sequencing can be valuable in that case. It's really key that you have the disease-relevant tissue or some other proxy cell type that you think is close enough that the genes of interest are likely to be expressed in that particular cell type, and that, of course, won't always be the case for many diseases. And in general, we would suggest running whole genome sequencing in parallel to RNA sequencing, because that means that if you find something very unusual in the RNA-seq data, you can go back to the whole genomes and see if there's a mutation that's present in that region, as we saw in our collagen 6 case. So that's all I wanted to say about the different genomic technologies today. Some points for discussion if there's time for any sort of chat. One question, of course, is at some point we know that genome sequencing will become the frontline technology. It clearly outperforms exome sequencing in a whole series of different ways. The big challenge, of course, is the cost differential. So while we have that 5 to 6 times cost differential between exome and genome sequencing remains much more appealing for most applications. And so I think it's still going to be another year or so, maybe longer, before genome sequencing becomes sufficiently cost-effective that we start running it instead of exome sequencing across the board. There's a second interesting question about whether there are particular classes of disease gene that we're missing with exome sequencing that we can only discover, or diagnoses that we can only make using whole genome sequencing compared to exome. And I think we don't have many great examples of those yet, but I'm sure they will occur. We have challenges that we wrestle with about how we should think about approaching gene discovery in the probably relatively large fraction of severe rare disease cases where there's not a single genetic cause, that is, there's not a single monogenic cause of that particular disease. So these might be things like oligogenic disease, for instance, where a particular patient has 2 or 3 mutations in different genes that in combination result in a very severe disease, but no one mutation is actually causal in that particular patient. And then a final question we wrestle with a lot, of course, is which tissues are the most relevant to perform RNA-seq in for any particular disease. So with that, I'll finish by thanking, of course, everyone who's helped to put these slides together. And once again, thanks to Sam and the team for putting this course together. I'm looking forward to a couple of days of really productive discussion with all of you. Thanks very much. Now, Sam, should we have the time for a couple of quick questions now before we move on? Did anyone have any questions about anything that's come up in this first presentation? Hi.
Thank you so much. You mentioned in the last case for RNA-seq that the patient did go through cDNA analysis and still that mutation was missed.
Yeah.
Can you elaborate?
Absolutely. That's a really great question that I actually skipped over briefly. So this previous cDNA analysis had actually been done in fibroblasts. They had taken fibroblasts from that particular patient and then grown them up in culture and then done cDNA sequencing on those fibroblasts. And in hindsight, looking back on the cDNA sequencing, you can see a tiny little blip in that particular region where that exon is found, but it's tiny, almost undetectable. And the reason for that is because this particular inclusion turns out to be almost entirely muscle specific. So because we did our RNA sequencing on a muscle biopsy, it shows up very clearly in the RNA-seq data. But in the fibroblasts, where typically collagen 6 is expressed at high levels, this specific inclusion doesn't occur. And that's just incredibly bad luck. It just turns out that this particular inclusion, just for whatever reason, must be close to a muscle enhancer. That means it's only switched on in muscle, and that explains the muscle-specific phenotype in these cases. Any other questions? Oh, great. Okay.
Hi, very nice talk. So you mentioned a lot of things about the weaknesses of short read sequencing. Are you guys exploring long read sequencing too?
Yeah, another great question. So many of you guys, I think, will know about long read sequencing. These are relatively newer technologies like Oxford Nanopore or Pacific Biosciences approaches that result in typically single molecule sequencing, but with reads that are much, much longer. In the case of Oxford Nanopore, potentially even hundreds of thousands of base pairs long. So the big advantage, of course, of those technologies is that you can imagine reading almost an entire chromosome in one read, and that would be huge. It would allow us to identify, for instance, all of the really difficult classes of structural variation would fall out very easily if we had those super long reads. The challenges of these technologies still are, firstly, they have much higher error rates than short read sequencing. So they have error rates typically substantially over 1%. So that can make it much more difficult to actually analyze the data that falls out. And the SNPs and indels you get from long read sequencing tend to not be as accurate. And the second big challenge for us is actually still the cost. So to do a whole genome now with Pacific Biosciences technology would cost us about $25,000, maybe a little bit more. That's to get a reasonable coverage whole genome. And so at this point, it's just not cost effective to go after those cases. I think perhaps only in those situations where we've really taken everything we can, we've done short read whole genome sequencing, we haven't been able to discover the cause, we think the family is really compelling and we just need to apply that next technology, maybe that would be worthwhile. But still, $25,000 is a lot of money to spend on one genome. As the cost drops, I think that will become pretty competitive, and we're certainly keeping a very close eye, in particular on Oxford Nanopore, where the cost decline seems to be going in the right direction, where eventually that may make it cost effective to do a combined approach where we do a very low coverage long read sequence of an entire genome to get those structural variation. And then on top of that, we also apply short read sequencing to pick up the high confidence SNPs and indels in the mappable regions of the genome as well. Great. If there are no further questions, I'll hand back over to Sam to introduce our next speaker. Thanks very much.